It shows lots of different plants, and roughly estimates there are about 400,000 plant species in the world. And these 400,000 plant species are being attacked by more than 1 million different insect herbivores. But clearly the world is still green, and that means that most plants are not eaten by most insects. And so why is that? And that has primarily to do with secondary metabolism or plant metabolites. So each plant has roughly 30,000 genes. It can be a few tens of thousands more or less, and about 5,000 metabolites. Out of these 5,000 metabolites, about 500 of these are what we would call primary metabolism, so things like sugars, amino acids, lipids, carotenoids, things that every plant has that make a plant a plant. And the rest of these, about 4,500, are part of specialized metabolism. And that's really what makes plants interesting, is that they're the chemistry of the natural world, and there's literally more than 1 million different natural compounds known from plants. But whereas all 30,000 genes in pretty much any plant are easy to find these days, genomes are sort of a dime a dozen, these specialized metabolites in most plants, we know that they're there because we see a feature on the mass spectrometer, but we don't know what these metabolites are, we don't know what they do, and we don't know how they're made in these plants. And that's really what's driving the research in my lab, is this specialized metabolism and how these specialized metabolites then provide defense against insect herbivory. So I'm going to highlight a few of the approaches used in my lab to study plant specialized metabolism and defense against insects. One approach is just general gene discovery or metabolite discovery. We look for new, previously unknown metabolites that are used for defense against insects. This here is an acetylated catechol glucoside, which Boas Neganen and Richter found in maize, so a previously unknown maize metabolite that's involved in plant defense. And we've done this for a number of different metabolites using the same general approach. We identify the structure of the metabolite. We find biosynthetic genes, usually by GWAS these days for plants like maize. And then we do bioassays with mutant plants to say, if we knock out this molecule, what does it do for plant defense? This is a new molecule, but it does have structural similarity to things known from spruce that provide protection against spruce bedworm. Another common theme in plant defenses is repeated evolution of identical or very similar molecules. What we have here is a cardiac glycoside, and cardiac glycoside biosynthesis has evolved at least 12 different times in different plant families. And so we're studying plant biosynthesis of cardiac glycosides because these are important defensive and also medically relevant molecules. And the main questions that we're trying to answer now is, how do plants make cardiac glycosides? That previously wasn't known. Do different plant species that have independently evolved these pathways evolve the same or different pathways to make identical molecules? And variations on this structure, there are several hundred different cardiac glycosides known in nature, but they all target the same enzyme in animal metabolism. And so the question is, why do plants have 500 different cardiac glycosides? And we're now able to make mutants and make different cardiac glycoside profiles to answer these questions. We're working, sorry, computer problems here. Specifically with two species, wallflowers in Brassicaceae and milkweed in the Apicenaceae for this research. And then finally, we've really benefited a lot from recent USDA regulations regarding CRISPR-mutated plants. We can now make CRISPR mutations and plant plants outside in the field and do insect studies. And here is an example of that sort of thing. We've been doing experiments with Nicotiana benthamiana using a protocol that Dan Voitas outlined yesterday, making mutations affecting nicotine biosynthesis, acyl sugars, and protease inhibitors in all these different combinations, planted them out here in a field on Freeze Road, and then just let the insects attack. And then we can determine what's the relative importance of these different defenses and whether some defenses are important against some insects or other insects. And as you might expect, the answer is sometimes yes and sometimes no. And then finally, I'd want to point out Layla is going to be giving a talk in the next session about none of these subjects. She actually hasn't told me what she's talking about, but it'll be an interesting talk. Thank you.